NOSB webinar series. I'd like to introduce myself quickly. I'm Melissa Verdor, and I work at the National Office for the National Ocean Sciences Bowl. I'd like to welcome you tonight to our second webinar. If you were lucky to join us in November, you saw Dr. Larry Krissick present, and tonight we have Dr. Brian Huber. Before I introduce him and have him start presenting on his topic, I just want to give you a few bits of information. Just to remind you, tonight during our presentation, you are all on mute right now. So if you have questions for Dr. Huber while he's presenting, there is a little questions comment area with the webinar system, and you can just type in your questions, and the NOSB staff here is going to field those questions and give them to Dr. Huber as appropriate, or we'll save them all till the very end. I'd also like to remind you that there is a technical help number, just in case tonight you have problems either hearing or seeing the presentation. There's not much I can probably do from my end as the organizer, but if you have any issues, please call this 1-800 number. It is 1-800-263-6317. Also, just to remind you that the last presentation and tonight's presentation, we are recording them. I made sure to hit record tonight. They are going to be posted on the NOSB website. So I'm going to go to the website right now. It's just nosb.org, and we have a professional development tab. If you click on that, you'll see the information about each of the presenters, and if you click on their name, it'll take you to a page where you can both watch the recording and also look at a PDF of the PowerPoint presentation. So tonight, like I said, we have Dr. Brian Huber. He is with the Smithsonian Institution, and he is going to talk about planktonic foraminifera and how and what they can tell us about the Earth's environment and how it's changed over the past 120 million years. So if you'll just bear with me 1 second, I'm actually going to give control of the screen over to Dr. Huber, and we'll make sure everything's working correctly. Oh, there we go. So we can see you now.
Yeah, hi, I'm Brian Huber. I'm a curator of a microscopic group called Foraminifera. I work at the Smithsonian Institution Department of Paleobiology. Right now I'm chairman of my department. I've been in the department for 23 years. My job is mostly research. I was just telling Melissa and Kathleen that I've worked on Ocean Hall for 5 years. It's a large exhibit that opened in 2008. And if you haven't seen it, I welcome you to come check it out. It's 24,000 square feet of stories on the oceans. And I do a lot of research in Cretaceous time, and so I'll be talking about geologic ages quite a bit. I'll give the ages for those time intervals that I talk about, but I realize a lot of you do not have a strong background or any background in geology, so I'll try to keep this at a level that's pretty accessible. Melissa, do you want me to just go ahead and get started, or what do you think?
Sorry, I had muted myself. I say go right ahead.
Okay. Okay. So the group I work on is called Foraminifera, and we're fortunate that this group of organisms is in the ocean today because we can study them, we can culture them, understand their biology, understand what their tolerances are to different salinity and temperature ranges, and things like that. On the left of this title slide, we have Benthic Ferminifera. They live in the seafloor. In this case, these are larger Benthic Ferminifera that have little photosymbiotic algae living with them. They grow very big. That's probably a couple millimeters in diameter there. These are foraminifera living in the reef, but we also have foraminifera living in the very deep sea, even in abyssal depths, in terms of benthic foraminifera. On the right is one species of living Planktonic Foraminifera. Planktonic or Planktonic, they float. In this case, you can see the spines arranged around the shell, and the spines have these little lights that are actually photosymbiotic algae living with this Foraminifera. Now, Foraminifera are single-celled protozoans or protistans. So they're special in that they secrete a shell, and these shells, of course, then leave a fossil record. This photograph actually shows a Planktonic Foraminifera capturing its prey. That's a little zooplankton that's caught in the spines and is getting ready to feed on it. So my talk is called Foraminiferal Evidence of Dramatic Climate and Ocean Change During the Cretaceous Period. Now, the Cretaceous is a period that spans from about 145 to 65 million years ago. I'm going to talk about three time intervals in particular for the Cretaceous period. But first I want to go over a little basics about the modern ocean floor. This is a map of the sediments of the ocean floor. And you'll notice the different patterns here. The brick-shaped pattern is calcareous ooze. And you'll notice that the sediment changes in different areas of the ocean. Calcareous ooze is stable at depths above about 3,500 meters. Below that depth, the ocean is too acidic for the calcium carbonate to be stable, so it dissolves. But calcium carbonate, the calcareous ooze, is mostly composed of the shells of Foraminifera and things called calcareous nanoplankton, which I'll talk about. Around Antarctica, you've got glacial marine sediments, so this is a climate indicator, of course, for times when you've got these glacial advances to get those sediments out there. And then beyond that, you've got siliceous ooze. This is from the shells of biosiliceous organisms like diatoms and radiolarians. You also have siliceous ooze in equatorial upwelling zones, such as here. You may already know about this. Where you have upwelling of nutrient-rich water, you get these siliceous sediments. And then big areas of the Pacific in particular have red clay, where it's too deep for calcium carbonate and there's not enough productivity for the biosiliceous organisms. So the point of this slide is that the ocean floor has very many different kinds of sediments, but the carbonate sediments are quite widely distributed. And for this record of the microscopic fossils that I study, we go after carbonate sediments in the geological record. Now, I'll just briefly mention calcareous nanofossils. Chalk is going to be full of these things. These are really, really tiny. Here's a scale bar, 1 micron in length. So these things are just a few microns in length. Typically, they form a sphere or sort of a three-dimensional object, but when they die and decay, they fall apart into these little platelets. So that's the predominant composition of calcareous chalk. So my group is Foraminifera. This is a typical random distribution of Planktonic versus Benthic Ferminifera. This is a Benthic down here. These are all Planktics here. Typically deep sea sediments, or that is sediments that might be below 1,000 meters depth, are mostly composed of Planktonic Foraminifera. So 95% are Planktonic Foraminifera. Shown on the lower right is a modern or living Planktonic foram. Again, here's the shell in the center. This is the protoplasm, and then the spine sticking out. And so these things are the floating ones. And then up here is a benthic foram. Okay, so the benthics are pretty rare. But what's nice is both of them can give you some really useful paleoenvironmental information. Benthics can tell you about depth of the ocean. They can tell you about oxygenation and lots of other environmental parameters at the deep sea floor. And planktics can give you an indication of what the climate was like up near the surface and what the oceanography of the surface water zone was like. So a big advantage of studying Foraminifera is first that they are very abundant worldwide. In a small amount of sediment, you're going to have tens of thousands, hundreds of thousands of specimens to study. So you can look at these and tell age, because each species has a limited age range in geologic history. So when you identify the level of first occurrence, That's a unique evolutionary event in geologic time, and of course its extinction is a unique evolutionary event. And like I said, they're sensitive to climate and ocean change, and so they're very useful indicators about Earth's climate and ocean history. For the distribution of modern planktonic foraminifera, I show this diagram where some species you see are most abundant, that is the diagram is widest here, where they're most abundant in the tropics, and then they become rare as you get toward the mid-latitudes, and they're completely absent from the high latitudes. Whereas other species dominate at high latitudes and they're absent in the tropics. Now there's a diversity gradient. Your highest diversity is in tropical, subtropical latitudes. At polar latitudes in the modern ocean, you only have 1 to 2 species. Total Planktonic foraminiferal diversity today is about 40 species. Now these distributions parallel the warm belts of the ocean, and that's what this map shows here. Planktonic foraminifera also have a depth distribution. Some species prefer to live in the upper water column, in the top 20 meters, maybe the top 50 meters, and others live deeper. And so all these species are arranged relative to where they can be found in the modern ocean. Now, a lot of my work is with Integrated Ocean Drilling Program, and particularly Deep Sea Sediments Core During Ocean Drilling Program, or IODP. And so I'll show you a few slides about that program. Right now, the main platform for drilling sediments where we reconstruct climate history is the JOIDES resolution. But we also have mission-specific platforms, some that go to the Arctic, some that drill on shallow carbonate platforms. And then the Japanese drill ship Chikyu is able to drill in very hard rock and drill in very shallow zones because it's got a riser drilling system, a different kind of technology than the JOIDES resolution. So look, most of my work, actually all of my work, has been based on either the JOIDES resolution or the Glomar Challenger sediments from the first 30 plus years of, of ocean drilling. So the resolution is a very fine ship, 470 feet long, and it has about 20 scientists who are there for the objectives of the research of a particular cruise, and then there's a bunch of science technicians that work with the research scientists for each cruise. Each cruise is about 2 months long. So it's capable of drilling great depths, and its core recovery can be excellent. It can be 100% for quite long intervals. So it's a very valuable research tool. When you consider the scale of this drill ship drilling in 2,500 meters water depth, it's pretty incredible that that length of pipe can go all the way down. They can drill, and as they drill, the core is advancing, and the drill pipe advances, and the sediment that's drilled comes out in a little hill outside the drill pipe. There's a apparatus that's called the core barrel that is sent down, and that has a tube that contains the sediment as we drill in core. This is the drill bit. This is the outer bottom hole assembly drill bit, drilling the sediments, and then this is the extended core barrel where we've actually got the plastic tube inside this, and as this advances, the sediment goes inside that tube, and then we send a cable winch to retrieve the cores as they are drilled. Here's a view from the top of the drilling derrick looking down. This is the helicopter pad. This is the pipe rack where all the pipe is stored, and this is the top drive, which is what turns the drill pipe as the drilling goes on. This top drive weighs, I think, I don't know, over 50 tons or something, maybe 100 tons. It has to stay with the swell. So as the ship goes up and down with the swell, this thing has to go up and down the derrick. So it's pretty incredible technology to witness when you're on board the ship. Just an example of what it's like when you're pulling pipe. This is the machine that unthreads the pipe. The bottom part stays attached. This part comes apart, and they pull it away. You can see that the bottom hole assembly is pulled away here. Here they've sent the cable down, they've retrieved the core barrel, they're pulling out the pipe, and here they're pulling out the tube full of sediment. That long tube, usually 9.5 meters long, is then put out here. It's marked so that we have a line to split it into an archive and a working half. And it's also cut into 1.5 meter lengths so that we can study those easily on the sediment table. Here's an example of a working half of a pelagic carbonate core, or calcareous ooze. You can see the little sample containers for paleomagnetics and for calcareous nanofossil work and for the Foram people taken from this core. During the cruise, the paleontologists on board get a sample from the core catcher, which is right at the bottom of the core. Those immediately go down to the paleo lab, and then we wash it up, and then we provide ages based on the determination of species of the microscopic fossils. The photograph on the right shows quite different colored sediment. This is actually mostly composed of biosiliceous organisms like radiolarians and diatoms. These two cores actually were drilled near Antarctica. The first one is in much younger age sediments. This is actually Pliocene in age, so just in the last couple million years. This core is about 64 million years old. So climate at this site changed from very warm with mostly carbonate-rich organisms to a highly productive type oceanography, very cold with mostly biosiliceous organisms. Here's examples of some of the biosiliceous organisms. These are radiolaria from a biosiliceous type core that I just showed. So we do this work on board the ship. We provide the information to the drillers and to the other scientists in terms of what the age is, so we know what kind of progress we have during drilling. There's lots of science that gets done on board. There's a lot of organic chemistry, inorganic chemistry that gets done. Paleomagnetics, we want to see if the magnetic field of the core is reversed or normal relative to present day. And you can see this is plotted in the graph here. So we can get real-time paleomagnetics, which helps with our age determinations while we're on board the ship. This instrument is called a cryogenic magnetometer. These are some of the paleomagnetic guys at work. Now I'm going to talk about the Cretaceous Age. The youngest stage of the Cretaceous is the Maastrichtian. And this is a paleogeographic map, or a reconstruction, for the early part of the Maastrichtian, and this is a time about 70 million years ago. The diagonal lines on this map represent ocean floor that no longer exists because of plate tectonics. All of this ocean floor has been subducted since the Maastrichtian because of seafloor spreading. So if we wanted to drill the Maastrichtian, we would not find carbonate sediments on any of these areas of diagonal line because the ocean crust is too young. So this tells you that as we go back further in time, we have less and less of the ocean floor to study. The relative colors here are showing sort of relative depths, with the lightest color being shallower depth and deeper as you get further away from the spreading ridge. The yellow dots are sites drilled during the early, drilled recently, but to core Maastrichtian sediments. During this talk, I'm going to focus on several sites. One is off of northern Florida, a place called Blake Nose, or Blake Plateau. Another site is Falkland Plateau, which is at about 58 degrees south paleolatitude. And then another site is Maud Rise, which is near Antarctica. Now I want to mention that on this map you can see that there actually were seaways through the continents because of higher sea level. So not only does climate change, not only does the position of continents change, but sea level also changes, at times flooding the continents. So we had a seaway separating eastern North America and western North America at various times during the Cretaceous. It also seaways through South America, Africa, and so forth. Well, just an example of how do we use microscopic fossils to tell age on board the ship. This is a sample or a core from Site 690, which is right here, 65 degrees south, near Antarctica. The core is Maastrichtian in age. This column represents drilling recovery. The black represents core or sediment that was recovered, and the white represents an interval that was not recovered because of problems with drilling. So sometimes we don't get 100% recovery. This long stretch of black, you can see we have really good recovery right through this interval. But anyhow, we do our sampling. All these little numbers here are where I took my samples from microscopic fossils to determine the age. Now I'm going to talk about a particular useful species for determining age. It's called the Benthic Ferminifera, shown on the left here. Now, as I studied the cores, I was able to determine that the first occurrence of Benthic Ferminifera was in the core 28. And so we were actually up here, and you can see it's got its first occurrence here, and then it goes extinct up at the top right at the end of the Cretaceous. That species is tied to magnetic reversals. We know that its first occurrence normally is within magnetic reversal 31R, which is shown here. So that very level below seafloor of core depth can be pinned to a very precise age because we're in the middle of Cron 31 normal. And that 31 normal is given an age that's quite precise. So this graph shows the first appearance datum, FAD, at two different sites side by side, site 689 and site 690. And the age determined from that first occurrence based on the paleomagnetic plots is at one site 69.86 million years ago, at the other site 69.92. So they're very good agreements side by side. And so that's shown here. This is age of the first occurrence in what's called a sedimentation rate curve. Now this curve is determined by these crosses, which is where the magnetic reversals occur. This all gets very complicated, but I just want to show this to let you know that this gives us a very powerful tool because we are able to give an age, very precise age, at any level along throughout the interval that was drilled. And of course, once you have age, you can reconstruct history, and you can talk about events at one site compared to another site. And this is a photograph of the species I was talking about, Miorensis. Okay, so it's wonderful to have this kind of good agreement between sites. It gives you great confidence that we've got very good age control. And so this is what we really go for all sites. It's not always possible to have such good age control, but this is what we try for. Now I'm going to talk about geochemistry, something called stable isotope analysis. For paleoclimate analysis, what is called oxygen isotope analyses is a very powerful tool for reconstruction of ocean temperature through time. Remember that we have both Plankton and Benthic Ferminifera. Plankton live in the ocean surface, Benthic on the seafloor. Now, if we can get these oxygen isotope values from the shells of these Foraminifera, we can compare the temperature of the ocean surface to the ocean floor by using Plankton versus Benthic Ferminifera. And that's what I'll talk about in just a bit. At the same time we measure the stable isotopes of oxygen, we also can measure, the mass spectrometer gives us values of carbon isotopes. I won't talk about those today, but they are very useful for other oceanographic indications of productivity and the flux of organic carbon to the seafloor. Now, for reconstructing temperatures, we first need to express our values relative to a standard. So these are these stable isotopes of oxygen that we measure. Oxygen 18 16 of the sample is compared to a standard, and we express this in per mil values relative to that standard. Carbon isotopes is the same thing. And then we use a temperature equation to reconstruct the values that are coming from our sample, and we plug those in here with their assumed values of the isotopic composition of the ocean, and use this equation, which was empirically derived by experiments of living organisms, modern organisms, and that gives us a temperature. So we can plug in values from the oxygen isotope equation here, plug them in here, but we have to make assumptions of this delta W, which is this value here. And for an ice-free Earth, we assume this value is minus 1.2. So we plug in minus 1.2 here, we plug in the oxygen isotope value here, and here, and then we come up with our temperature. So a very powerful tool. And as long as preservation is good, this is going to give us temperature history through time. So I did mention preservation. It is really key that we have material that is well preserved if we're going to get the most reliable oxygen isotope temperatures. On the upper left, I show what is called a pristine shell. This is actually a shell that's 104 million years old, but it is perfectly preserved. It's a broken shell of a plankton foram. You're looking at the inside of the shell chamber. There's no secondary calcite. The pores are completely clean. There's no recrystallization of the wall. And so you can completely trust the chemistry that comes from this shell. This shell is also really well preserved. It's got a little junk in it. All you have to do is clean the sample, and then the shell is going to be very reliable for the isotopic temperatures. Now the shell in the upper right has been recrystallized. You can see that by this rhombohedral calcite growth, these little crystals that have grown on the inner chamber surface. This still is going to give us good values. It's not going to give us as reliable values as you would get from a shell like this, but it's still going to give us a good relative estimate of temperatures. The shell in the lower right is completely filled with calcite. So it is solid calcite. There's more mass of calcite inside the chamber of this shell than the shell itself. So actually, most of what you get from something like this would be a secondary signal, which would not be something you'd rely on for any kind of reasonable interpretation of climate change. Okay, so here's a curve that is reconstructed for the last 115 million years. And it's a curve that's from analysis of Benthic Ferminifera that lived at middle baffle depths. So between 1,000 meters and let's say 3,000 meters water depth. So the nice thing about analyzing Foraminifera through time like this is that you're getting a very good estimate of kind of the global climate condition through time. And if we start in the modern at 0, you can see we've got very enriched oxygen isotope values. The more positive these values are, the colder the temperatures. The more negative they are, the warmer the temperatures. So let's go back in time. Now in the Neogene, we have what is called the mid-Miocene ice buildup event. That's right here. Look at the sudden drop in oxygen isotope value, or actually increase in oxygen isotope values, which means a cooling of oxygen isotope values. This is when a huge ice cap grew in Antarctica. Go back further in time, and you see a few wiggles in the Miocene, and then we get into the Oligocene, and we see a big event that was a warming event as we go from late Oligocene into the Miocene. Go back further in time. Look at this big step here. This is a big cooling step right at the Eocene-Oligocene boundary, 34 million years ago. This is when major ice sheet first appeared in Antarctica. And so these deep ocean benthic foraminifera are very sensitive to that climate change, and you can see a very dramatic increase in the oxygen isotope values, very big cooling event. Go back further in time, we got a very warm, deep ocean in the Eocene, early Eocene in particular, and this is what's called a Paleocene-Eocene thermal maximum, which is a big warming event. And then we get into the Cretaceous, 65 million years ago. And these data here were generated by a colleague of mine named Jim Zakos and his co-authors. These data here were generated by myself and my collaborators. And so we've extended this Cenozoic benthic isotope curve all the way back to 115 million years. And look at this. Now we had warm temperatures in the early Eocene, but compare that to these temperatures. The deep ocean was extremely warm. And in fact, at one point, middle baffle temperatures reached as much as 20 degrees centigrade in the early Turonian, which is phenomenally warm. Now at this time, the polar latitudes were also very, very warm. We've got isotope temperatures from Antarctica, near Antarctica, that also get really high temperatures. So we have a huge amount of climate change during the last 115 million years. And you can just see that by this beautiful benthic isotope curve that's reconstructed from the deep ocean. I'm going to talk about several time intervals. One of them is in the super greenhouse, which is this really, really warm period that I talked about. The first one I'll talk about is actually at what is called the Aptian-Albian boundary in about 112 million years ago. So that's right here. You can see that on this temperature curve, relative to other Cretaceous temperatures, this is pretty cool deep water temperatures. I'll talk about the sedimentary boundary, which is right here, very, very warm time. And I'll talk about the late Maastrichtian warming event, which is right here, at about 65.8 million years ago. Well, here's a paleogeographic reconstruction, actually for about 90 million years ago. And so this is a little bit older than that early Maastrichtian reconstruction map that I showed. Notice the seaway across North America is much wider, much broader. So this is about the highest sea level that Earth has had in the last 250 million years. So much of the continents were flooded worldwide. Notice that the South Atlantic is almost closed, that there's almost no circulation between the South Atlantic and the North Atlantic at this time, 90 million years ago. I'm going to talk about these sites, and this pristine preservation is really key, because if I want to reconstruct temperatures, I want to get the best preserved material. And unfortunately, not only do we have the problem of subduction taking away the seafloor, but we also have the problem of much of the sediment of carbonate that's preserved is not preserved well enough for reliable temperature estimates. If it's limestone, you cannot work with it. Even chalk, you can't get the most reliable temperature estimates. But these are sites that I'll talk about where we do indeed have some very nicely preserved material to work with. So this first interval that I wanted to talk about, Aptian-Albian boundary event. This is 112 million years ago. And the boundary in this core is shown right here. It's actually right at the bottom of a core section. But notice, so this is the top of this particular core, and then each of these connects up. And so this top here connects to the bottom here. So we go from quite a dramatic color change from this white to this gray. And this is right where the boundary level is. And I determined this based on studying the Benthic Ferminifera in the species that are present. Also in this core is this very dark laminated interval. It's called a black shale. And for this particular interval, it's called Oceanic Anoxic Event 1b. Now this is a weird time when the ocean bottom lost its oxygen. It was dysoxic. Not completely without oxygen, because there's little Benthic Ferminifera that live there, but they're tiny, very thin-walled, barely alive. So it's laminated because you don't have bioturbation, because there wasn't enough oxygen for the animals to live in the seafloor and mix up the sediments. Now, this is a really interesting time, because this black shale was thought to have formed under quite warm conditions. So my goal for this core was to obtain Foraminifera from these samples in the core below and above, and reconstruct the temperature through time for these cores. So I'm going to show some of those results. But the first thing I had to do is determine the age of these cores and determine whether or not the sequence was complete. Well, in quite a long time it took me to study this core. I had to do the taxonomy and actually name several new species. And I discovered that, in fact, there's a very abrupt extinction right at that boundary level, right where that color change is. You can see there's about 12 species here, Aptian age species. You go right up to that level where that color change is, and they abruptly go extinct. Then, just above that, you only have 2 species present, until here you start to get a couple more species coming in. On the right, you can see that I have a shell diameter plot. And there's a very abrupt change in the shell diameter, going from larger size forms to much smaller size forms. I also have a plot of percent planktics, and that's relative to the benthics. And you can see that the percent planktics actually drops right at that boundary level. So that was really interesting. What the heck is going on there? Well, to show the Foraminifera to scale, to just give you a sense of how dramatically the size changes right across that boundary level, I show the assemblage from the late Aptian compared to the early Albian, where there are just tiny things in the early Albian and these huge things with very distinct wall textures in the late Aptian. And you can see the meters below seafloor. This is just separated by 5 centimeters between where these 2 samples were analyzed. So very dramatic extinction. It's about 80% extinction of the assemblage. Now I've analyzed the oxygen isotope values and the carbon isotope values right across that interval. Now this is a very busy graph, but here's this level where the color contact is. And what I see is first a very, very big change in the carbon isotopes. In fact, here you can see that it's a 2.2 per mil shift, which is huge for the ocean, right within a centimeter sampling interval. At that same level, we have a big shift in oxygen. They actually get more negative, which means warmer, right there. Now, because this occurs at a sharp lithologic contact, my suspicion was that there was nonconformity here, meaning there's time missing. And with further study of this sample, I realized that there's at least 1 million years missing right at this color contact. Nonetheless, we've got a beautiful record of, well, let's concentrate on the oxygen isotopes. A beautiful record of the benthic oxygen isotopes, which are the diamonds, going right across this interval. And then the Planktonic Ferminifera are shown by the other color patterns. And here we can see that the Planktonic values are quite close to the benthic values, except for where the black shale is. That's where you have the warmest temperatures of the planktonics of the surface ocean. But you can see that the ocean bottom really did not change much across that interval. So this is sort of helping us sort of reconstruct what's going on in the ocean. It doesn't tell us necessarily exactly what caused the extinction, but we do see very significant change. What's interesting is when you use the temperature equation, the deep ocean temperatures are quite cool, and what's really surprising is the plankton values are also very cool. And so even though this site was at about 25 degrees north at this time, the temperature estimates are on the order of 12 degrees centigrade for the Aptian, and the warmest they get is close to 20 degrees centigrade during the early Albian, but mostly sticking around 14 degrees C. Present day in the subtropics is typically about 24 degrees C, somewhere around there at this latitude. So this was much, much cooler than we expected, and quite surprising result. So to explain that, here is the location of the site, plotted on a map 108 million years ago. This is a very restricted North Atlantic, and we think that the North Atlantic was more saline, and salinity actually does affect your temperature calculation. The higher the salinity, the cooler the values will suggest. But you might have very warm ocean, but if it has high salinity, the oxygen isotope values are going to indicate or wrongly suggest that you have quite cool temperatures. So we think that the North Atlantic basin at this time was much more saline than present day, which is what would explain the very cool temperature estimates. So notice that there was some kind of, not necessarily landlocked, very shallow a bridge of raised seamounts or volcanic arc in the western part of the North Atlantic. So that restricted the water flow from the Pacific. Also notice that we probably had saline water being evaporated from the shallow seas of the North America and also from the shallow seas of South America. So very high evaporative seas contributing high salinity to this North Atlantic basin. So much different North Atlantic at this time than what we might consider in much younger time. Well, going to this site that's off of Falkland Plateau at about 58 degrees south in the South Atlantic, we actually find some cores that do not have an nonconformity. And in fact, we do see the same extinction that we see at the site in the North Atlantic. Very big drop in the percent planktics and a huge shift in the size of the plankton forms to tiny forms, just like we saw at the other site. So we know that there's a huge extinction that goes on here. We know that there's big change in ocean chemistry. What we don't know is what the cause was, and this is something that we're investigating right now. The oxygen isotopes and carbon isotopes from that site that I just showed you, so from the site 511, are shown here, and again we have a huge geochemical shift right at the extinction event. So a big event. All right, so now I'm going to jump up in time. We go up to 94 million years ago, and this is a reconstruction again for the 90 million years ago time period, showing site 1050, which is what I'm going to talk about. It's right off northern Florida again. And again, we have a big extinction. There's several species that are very prominent for millions of years below this level. They go extinct right here, and whereas other species go right across that same interval. This is a photograph of the core at site 1050 from the Sedimanian across to the Turonian time. So it goes extinct right in the top of the Sedimanian. At the same time that this happens, we see just above the extinction evidence of major volcanism. And we've tied this volcanism to the Caribbean large igneous platform eruptions. So there's a big igneous platform that was forming in the Caribbean Sea at this time, and we think that this extinction is related to those large igneous platform eruptions. Now, the stable isotope curves from that same interval, here's that same core. We do have a carbon isotope shift right at that time. But most importantly, what's really dramatic here is the diamonds. Those are Benthic Ferminifera, living probably 1,500 meters water depth. They're giving values of 19 degrees C, really, really warm. And in fact, those values are right together with plankton values. The species that went extinct are shown by the X's. They were the deep-dwelling planktons. And our hypothesis was that the ocean surface and ocean bottom warmed so much that there was no longer a significant thermal gradient and no longer a thermocline that was probably an environmental signal for these things to reproduce as they get to adult, as they grow into the deeper water. So this species, Rotellapora, which is shown here, went extinct probably because the structure of the ocean water column completely changed with this deep ocean warming. At least that's our hypothesis. And so the next site that I'm going to show is site 690, and I'm going to jump up in time. I should have changed that. That should say Maastrichtian. Sorry, I meant to change that subtitle. So now in the Maastrichtian, between 65.9 and 65.6 million years ago. Now here's another event that wasn't a big extinction, but in fact what we found was oxygen isotope values got more negative, which means they got warmer. These diamonds are all Benthic Ferminifera. The circles here are also Benthic Ferminifera. But especially the Planktonic Foraminifera got much warmer. This is a site that was at 65 degrees south near Antarctica. And so these values are warming to on the order of 15 degrees centigrade, so really warm for this brief amount of time. Now there's another site that was studied in the North Atlantic off of New Jersey, and the same thing happens. You can see that the oxygen isotope values of planktons got very warm at this very same time. Now what's interesting is this bug, this foram that I show here, is the same species as here. The only time I ever see this in Antarctica is right during this warming event. It actually evolved and existed way down here in lower latitudes. But it was only when the high latitudes got warm enough that it migrated to high latitudes and was present for a very short time during this brief warming event. So we've tied this warming event to actually a volcanic, big series of eruptions called the Deccan warming event. Deccan troughs are off of India, and there was a huge amount of igneous basalt that was erupted right at this very time. And we think as a result of that volcanism, we had a big global warming. And evidence for that volcanism is shown by osmium isotopes. And so this very same time interval, we can see the osmium isotopes have a huge shift as an indicator of volcanism. Okay, so the last few minutes of this presentation, I'll talk about the Tanzania drilling program, which is something I've worked on for the last 5 years. This is where we actually take a drill rig, shown here, and the drill rig is on skids. It comes in a big truck. We drill sites down to about 150, 200 meters to core sediments that used to be in the deeper ocean. So in this case, probably 200 to 500 meters water depth. But they've been gently uplifted in East Africa, and so we can access these on land. Now, working in Africa on land, of course, is far different from working on the drill ship. Fortunately, we had colleagues in Tanzania that we work with, and here's a colleague shown here from the Tanzania Petroleum Development Corporation. We also were able to use their vehicles and several of their geologists. The roads in Tanzania are not the best. Sometimes we got stuck. Sometimes there's large rocks, which blows lots of tires on the trucks. So there are logistics complications in doing this work, but it is quite exciting work because it's an area that has been unexplored up until when we were drilling there, and we made a lot of very interesting discoveries. So our goal for this Tanzanian project was to reconstruct Cretaceous climate history. There's various hypotheses that we want to test. I won't go into those in detail here, but basically we're looking at climate change on long and short time scales. We're also looking at changes in ocean chemistry. So the location of our area of drilling is where this black square is in East Africa, Tanzania, southeast Tanzania actually. And we drilled in two regions. One is called Kilwa and one is called Lindi. These are little villages. Shown on the right are the age intervals of all the sites we drilled between 2007 and 2009. And so a lot of our work was in the Tronian, this time of the greenhouse warming. And so you can see lots of sites drilled there. But we also have very good coverage for a bunch of the other Cretaceous intervals. This is age right here. So just wanted to show you what it's like going down there. This is the drill rig coming on a big flatbed truck. And then they get it off the truck on skids. And this is showing them offloading the drill rig from the vehicle. And then here it is, they've got it on site and they're jacking it up, leveling it, and getting it ready to drill. Now extracting core from this is a lot different than what we have with the drilling ship. This is far more primitive. Basically they're beating the pipe and getting the core out, and we're using them, putting them out on these tins. And then we have a measuring stick and we measure each length of core that comes out. So the drill is being extended down and they drill deeper and deeper, getting into older and older sediment. Here's an example where they drilled all night while we were sleeping. It was dark, so we couldn't work. When we got there the next morning, this much core was waiting for us to describe. This is our chart showing the cores and the drilling depths and the amount of drilling recovery for each core, which then we put into the laptop, which is powered by our solar cells. Here's an example of the core that we recovered with the measuring stick next to it. Beautiful laminated sediments in some intervals. Our goal working here was actually to work on sediments that were beautifully preserved or contained beautifully preserved foraminifera. And in fact, much of what we recovered did indeed contain gorgeous foraminifera. And what we ended up with is a very nice, very detailed curve that, for oxygen isotopes that spans between about 93 million years ago to about 89 million years ago. These are all Planktonic Ferminifera on the top. These are all different species of Benthic Ferminifera. And what's remarkable is just how steady the temperatures are throughout this entire time interval. Very little climate change at this time. Now this is interesting for us because some people have hypothesized that there were glaciers in Antarctica at this time. And if there were glaciers, we would expect that the isotope values would be shifting because ice contains more oxygen-16 than in the ocean. And so we would see these values shift, both benthic and planktic shift. Because they don't shift, we can pretty confidently say there was no ice in Antarctica at this time. So it's quite a successful drilling season. We've got a lot of other science coming out of this, but I thought this would be a good opportunity to show some examples. Here's a photograph of the beautifully preserved, one example of a planktic frame from the measurement. This is a benthic here. And here's another benthic that we measured in a third. Okay, so this is one of the drill sites in Tanzania that we ended up with. And with that, I'll say asante sana to the PowerPoint part of my presentation. Asante sana means goodbye, or thank you very much. And I guess at this time, Melissa, if you want to chime in and let me know if there's any questions, I'd be glad to talk.
Hi, Brian. This is actually Kathleen, and we've had a couple questions that have come in. There was one that came in from earlier. Are the Foraminifera fossils, or are some from other non-fossilized remains?
Sorry, could you repeat that? I don't understand.
It says, are the Paleoforaminifera fossils or some of the other non-fossilized remains? So I guess what they're trying to ask, and if the person who wrote this question, if I don't get it right, please feel free to write it again. But what I'm understanding is, are there any fossils that you're finding, are they all Foraminiferas, or are they potentially maybe some that aren't fossilized? Does that make sense?
Yeah. So for these deep sea cores, the ocean drilling cores, I am focused on study of Foraminifera, but the samples themselves are full of calcareous nanofossils. And in fact, there's other microfossils as well, like ostracods. They're quite rare. There's also occasional calcareous dinoflagellates and some other kinds of fossils, but you do not see clams or snails or things like that because we're really too deep for those kind of invertebrates.
Have you found Foraminifera fossilized in basalt?
No, Foraminifera do not occur in basalt. They only occur in sediments. Basalts are igneous rocks, and they form from the cooling of magma, whereas Foraminifera are the shells of organisms that lived in the ocean. Once the organism dies, the shell sinks to the seafloor and they accumulate at the seafloor. So they occur in quite different situations.
Another question is, did you say you determined age by something like index fossils, or do you use radioactive dating?
Good question. The example I gave you for biostratigraphy, it was using index fossils. And so I showed that species called Mayorensis, which is really useful as an index fossil because it's very widespread, it's very distinctive, easy to identify. And its first evolutionary occurrence is quite synchronous, meaning it occurs at the same time worldwide. So when you see that fossil, you know what the age range is because that species evolved at about 68.5 million years ago, and then it went extinct at the end of the Cretaceous 65 million years ago. And so we determine ages first using biostratigraphy and the species of Foraminifera and calcareous nanofossils. We also use paleomagnetics when we have that kind of information available. It's pretty rare that we can use radiometric dating. You need particular minerals like zircon or a particular kind of biotite or feldspar in order to get radiometric dates. In these kind of chalk settings where you get this carbonate ooze accumulating, it's oftentimes very, very rare that you're going to find any of these minerals that can be used for radiometric dating.
What aspect of the deconvocalism are thought to have caused global warming? We often read that volcanic activity leads to cooling by ash and sulfur dioxide entering the atmosphere.
Yeah, that's a very good question. Certain kinds of volcanism can cause an ash cloud to extend globally, but particularly it'll cause the high latitudes to warm because of a greenhouse effect. And so the effects of volcanism will vary depending on the kind of volcanism. Deccan volcanism is a basalt that was not very violent kind of eruption. It's Hawaiian kind of volcanism with gentle ash clouds. And apparently the ash and the dust that forms from that is a kind that causes warming, particularly at high latitudes. And so that's thought to have been the cause for this global warming at the end of the Maastrichtian age.
I would like to ask if you have found pollens and spores in those cores in, I'm going to pronounce this incorrectly, sorry, Turonium age, and if anyone has studied them. during the Turonian Age?
Turonian Age cores that are from the deep sea, the chalks and ooze, typically do not contain pollen and spores. Pollen and spores have organic walls, and oftentimes in the calcareous sediments, the organic matter will decay or oxidize away. So it's oftentimes you cannot find any dinoflagellates or pollen or spores in chalks. But in the Tanzanian material, yes, indeed, we do have pollen and spores. We do not have a colleague working on pollen and spores right now. We've got people working on the other microfossil groups, but so far we don't have anybody involved with pollen and spores in that. And we would like to have them involved.
Why do anoxic black shales form, and what happens environmentally to cause a rapid transition to prolonged anoxic conditions in an area?
Very good question. So that black shale that I showed at Site 1049 was in an interval called Oceanic Anoxic Event 1B. Now 1B and the other oceanic anoxic events like 1A, 2, and 3 were all thought to have formed under quite warm, deep ocean conditions. The oxygen isotope temperatures that we got, as I mentioned, are much cooler than we expected. Much cooler than anybody expected for black shales to form. The current hypothesis for these black shales is that they'll form under high productivity conditions even if you have cooler, deeper waters. So as long as you have so much organic matter being formed up at the surface, as that organic matter sinks, the oxygen is used up in the process of trying to decay the organic matter. And so in that process of using up the oxygen, then the depletion interval forms what's called an oxygen minimum zone, which grows across the seafloor. So you might have quite a vertical thickness of dysoxic or anoxic ocean water as a result of high productivity. Another hypothesis for black shale formation has been ocean stagnation. It was thought that with a really warm ocean, particularly for the Cretaceous, you did not have much circulation, and therefore you did not have much oxygen mixing. A lot of our studies in the last 10, 15 years have shown that, in fact, deep ocean circulation was still quite vigorous, no matter what the temperature was. It's really the crucial factor is how much productivity there was, what the deep water temperature was, and then how much, how rapid did you have ventilation at particular sites. And so it can be very complicated. And when we originally, or when scientists originally thought that all the black shales formed under the same conditions, now we realize that each of these black shale intervals is quite unique, quite different. And so we're having to study them quite in a lot of detail, doing a lot of geochemistry, including organic geochemistry, to try to understand what's going on. So there's still a lot that's not understood. What we do know is it's a lot more complicated than we thought.
You show a curve with your data and the Zachos data. I was wondering how well do they match right at the end of the Cretaceous?
Well, in fact, Zachos et al. Data went back to about 68 million years. Our data go up to 65 million years. So there's a 3 million year overlap. And in fact, those data overlap right on top of each other quite beautifully. So it's it's a nice data set that merges, and I think it's an example of showing the power of oxygen isotopes and telling us the sort of as a thermostat of the Earth for the long-term geologic history. Very reliable. And so the Cretaceous curve that I showed is not yet published, but we are working on a manuscript to get that out. And so we're going to publish our curve with the Zachos curve and talk about just how extremely warm the Cretaceous was. And that by itself is a big mystery and very problematic for climate modelers. to try to understand how can we have an Earth so warm, how can we keep the polar latitude so warm, and how was it sustained? And, was it driven principally by volcanism or some kind of source of CO2, or just what was the major driver? It's a big question still.
One of the questions is there a hard copy of your research? And you just mentioned that some of the information hasn't been published yet. Is there any information that has been published based on your research, or is it all, we all kind of waiting for that to come out?
Oh, there's a lot of things that have been published. One of the papers I cited in this presentation was a paper that's in Geology 2002. I'm first author. I don't have the reference in front of me, but it's the journal called Geology. And Huber et al., if you look in 2002 and do a search, you'll find that paper. There's another paper published in Journal of Foraminiferal Research where we published the oxygen isotope data for the Sedimentation Extinction event. That was published in 1999. So again, I'm first author on that. The Tanzania data are what's just coming out. The data I showed, we just have finished up in the last couple months, and so we are working on a paper right now for that. And there's other data that I presented that also have been published, and perhaps I could provide a bit of a bibliography if that would be helpful.
Actually, this is Melissa. I was going to chime in and say anything you can get to us, we will put up on your page on our professional development site. But I was going to ask, is any of that research listed on your website through the Smithsonian?
Yeah, I'm on the staff webpage, and I probably haven't updated that webpage for a couple of years, but a lot of that stuff should be there. So anything, prior to, I guess, 2009 should be on that website.
Okay, tomorrow morning I will try to add the link to your website.
Good. Okay. Yeah, that'll help. So yeah, I could provide some PDFs for this, and I also have published on the Cretaceous Tertiary boundary extinction. The Benthic Ferminifera suffered over 90% extinction as a result of the asteroid impact. And I've got several papers on that. And if people are interested in that, I can provide PDFs of that as well.
Great. A couple other questions that we've had come in. Is anyone looking at the organic dinoflagellates from your samples?
Not the organic ones. We have somebody working on the calcareous dinoflagellates. They're called bithanellids from the Troni and Tanzania sites. But the only samples where dinoflagellates might come from would be the Tanzanian samples. And as I mentioned from a previous questioner, we have not analyzed any of the organic microfossils yet. We don't have a specialist working with us. We'd be glad to have somebody come aboard, but so far we don't have anybody.
Do you see any noise due to the KT meteorite?
Do I see any noise?
Yeah.
I'm not sure what this is.
If you wrote this question, if you could kind of provide a little clarification, we can try to re-ask it. Another one is, what do Foraminifera eat and why?
Well, they eat to live, so that's easy enough. Foraminifera are primary consumers. They are consuming things like copepods. Probably in the Cretaceous they ate dinoflagellates, maybe diatoms. So they're eating little zooplankton, maybe some other small protistans. And they're just passive floaters, but they're, in the Cretaceous they actually did not have spines, but they extended their protoplasm into these long filaments called rhizopodia. And those filaments were very sticky. And as they passively float along, some other swimming creature like a copepod might have swum into the rhizopodia and gotten stuck. As soon as that happens, those rhizopodia retract and bring the organism into a digestive vacuole. And I had the good fortune of actually working with a microbiologist colleague in Curacao. And we would go scuba diving and collect living foraminifera off the coast of Curacao, bring them back to the lab. We kept these culture vessels at a normal marine temperature, and we could then put the specimens we collected into these culture vessels and then feed them once a day. We fed them little shrimp nauplii. And we could watch as they would capture a little shrimp, baby shrimp, and then we'd watch as they digested them. And then we would continue watching until they underwent reproduction. So they're really complicated little things. They're magnificent things. And believe it or not, I can only identify 6 different species, but some people can identify about 12 species while scuba diving. And it's because even though their shells are 0.5 millimeter in diameter, the spines extend them out to a couple millimeters, and with the sunlight you can see color and you can see arrangements of the spines or the rhizopodia, and you can identify distinguishing characteristics that tell you which species you're collecting.
How do you determine latitudes when reconstructing the continents and their drifts from hundreds of millions of years ago, and how far back does the rock record show position of the continents?
Well, good question. There's several people who have these paleogeographic reconstructions accessible on the internet. Some of the reconstructions I use are from Christopher Scottese, and so if you just type in Christopher Scottese in Google, in paleogeographic maps, you'll come up with his website. You'll see these reconstructions. And in fact, he's got them going all the way into the Precambrian. And so, to maybe 800 million years ago, or 600 million years ago, I'm not sure. The uncertainties, of course, become greater as you get back further into time, particularly back into the Cambrian and older, because much of the land masses have changed so dramatically during that time that it's hard to know just what areas were above water and where they were located. But to answer your question about paleolatitude, by using the magnetic measurements of rocks from continents and sediments or igneous rocks in all over the world, you can look at the inclination of those magnetic minerals and determine where was North Pole based on those inclinations. And so through the very detailed paleomagnetic studies of these minerals, scientists are able to reconstruct what is called the paleopole position. On top of that, of course, we have the spreading ridges and the magnetic lineations that parallel the spreading ridges, and we can then sort of follow those magnetic lineages, and we can trace the ages of these ocean crust around the spreading ridges and determine the geometry of the ocean basins through time. And of course, once you get back into the early Cretaceous, Jurassic, you're running out of ocean crust to do this with, and so you are left with, as we get into the Triassic and older, just studying continental rocks to reconstruct these paleopole positions and positions of the continents. Of course, determining where the oceans were in the continents is simply a matter of mapping where the sediments were of that age. And as an example, in Kansas, we've got Kansas chalk all over the place. You can find mosasaurs, you can find ammonites, clams, all kinds of things right in the middle of Kansas. Well, what does that tell you? There was a big sea wave going to Kansas, and you can follow this sea wave with those same sediments through Colorado and up into Montana and on up into British Columbia during the mid-Cretaceous.
One of the questions came in, it's asking, based on your research, is global warming real?
Yeah, well, that's not my own research, but I'm very familiar with research that it is based on, and one of the most dramatic things that was done during my professional career was coring the ice caps in the North Pole, Greenland, as well as Antarctic ice cap, and even mountain or alpine glaciers. And from these ice cores, you can get a combination of carbon dioxide that are in the little gas bubbles of the ice, along with oxygen isotopes, and reconstruct the temperature through time together with carbon dioxide. And what you see is a record that goes back 150,000 years or greater, right up to the present. When you get to the Industrial Revolution, you can see once we get into the middle 1800s, late 1800s, the amount of carbon dioxide starts to pick up. By mid-20th century, the carbon dioxide picks up quite dramatically. Parallel to that, you can see the temperature rise as well. And of course, toward the end of the 20th century to present, we see a very dramatic rise in both of those measurements. So we know, based on climate modeling and our understanding of how the climate system works, that carbon dioxide is a greenhouse gas, and with increased carbon dioxide, we get warming. One of the things I often make people think about is consider that oil, natural gas, and coal has been in the earth for millions of years, and in some cases several hundred million years. In just the last half century, think of the billions and maybe trillions of tons of carbon dioxide that's been released to the atmosphere as the result of burning of these fossil fuels. That's a huge, huge amount of carbon dioxide dumped into the atmosphere. Now, given that carbon dioxide is a greenhouse gas, of course you can expect it's going to have a major effect on climate. So there's, almost no scientists now doubt that there is a link between carbon dioxide and temperature, and that in fact both have been rising during the last 2 centuries. The question that gets very sensitive is what to do about it, and whether we try to ameliorate this by major investment, which has a huge economic cost. The reality is, as we get, we're past 7 billion people on Earth, we all want our comforts, we all want to be able to drive around, and the most convenient way to do this is by burning fossil fuels. So there's no easy or good solution in sight. But the message is that anywhere you can save on burning fossil fuels, it's your responsibility to do so, because we're all contributing to this effect.
You referenced the influence of Caribbean volcanic provinces in the CT interval. What do you think was the response of the Plankton Foraminifera in the northern South America?
Well, in fact, the extinction that I mentioned at the Sedimentation boundary interval is global. So everywhere we drill, we see this major group of foraminifera called rotellaporids, along with a couple other species, go extinct at the same level. And we see also from measurements of the oxygen isotopes in the deep sea drilling sites worldwide that this deep ocean warming was global. So not only in northern South America, but everywhere, we had this rapid increase in temperature, which we think caused a collapse in the vertical stratification of the ocean, which we think then led to the extinction of this big group of Benthic Ferminifera.
How would this index fossil and relationships with climate be related to climate changes related to the extinctions caused by meteorite impacts, such as with the dinosaurs?
Well, fortunately, that extinction event is really well studied. And there's some beautiful deep sea cores on exhibit at Ocean Hall, and there's a number of sections worldwide where this record is very complete. And we can answer that question with pretty good confidence at this point. Well, we know, because of the mapping of the location where the asteroid hit, we know that the crater rings were 180 kilometers in diameter, and from that we know that the asteroid had to have been about 10 kilometers in diameter to form such big crater rings. Now, from modeling that, the asteroid was probably traveling on the order of 30,000 kilometers per hour, and that energy was so great that it vaporized crust where it hit. And the location of where it hit is Chicxulub in Mexico, Yucatan Peninsula. The amount of debris, very fine, volatile material that vaporized where the asteroid hit, shot up into the atmosphere. Along with that, the asteroid hit in a location where there were what are called evaporite deposits, particularly gypsum. Gypsum is calcium sulfate. That sulfur also went up into the upper atmosphere, and that caused shading along with all the dust from the ejecta where the asteroid hit. Plus the heat energy caused wildfires and a tremendous amount of soot. All of this contributed to darken the Earth's atmosphere to the point where very little or no sunlight got through for weeks to maybe months and maybe longer. And of course, once you cut the sunlight, you immediately shut down the food chain. All the primary producers collapse because they can't photosynthesize. And then the primary consumers, like the Benthic Ferminifera, no longer have their source of food. And so in the ocean, the food chain collapsed. And this is why the Benthic Ferminifera suffered such a huge extinction. Benthic Ferminifera, on the other hand, did not suffer much of an extinction because they are detrital feeders. They are able to find organic matter in the sediments and feed on that. So they could sort of hunker down, and even though there wasn't much food for a little while, they had plenty to eat down on the seafloor. On land, of course, the plants died, but a lot of plants have seeds, so eventually things germinate. But what we find in the land record is a huge spike in the amount of fern spores in the most complete sequences on land. And so where the dinosaurs become extinct, we also see a big plant extinction or disappearance of plants. Immediately after that, we see this huge spike in the amount of ferns, which suggests these opportunistic ferns were the first to come back and dominate the landscape. And so then soon after that, you get a recovery to the more normal kinds of vegetation. When you consider the kinds of dinosaurs that were around, of course, you had the herbivores feeding on the various, browsing on leaves or browsing on whatever grass-like things were there to feed on. Once those are gone, the carnivores came in. Mammals probably survived because they were typically smaller, possum size or smaller. A lot of them burrowed, and they probably had a stash of food. And so they were able to sort of, again, sort of hibernate or hunker down and survive through this very difficult number of months while the atmosphere took a long time to clear. So this is based on a lot of study worldwide, and the story is very tight now. It's very clear we have just one ejecta bed, and that ejecta bed is bounded by the record of Foraminifera, showing 90% extinction, and then just above it, a very rapid diversification from just a few species that have survived. So we no longer consider volcanism as a viable hypothesis for this big extinction event. Clearly, the asteroid was the cause for the global extinctions on land and in the ocean.
Let's see here. How has the evolution and changing of the oceans affected the samples of Foraminifera? And based on seafloor spreading, what do you predict will happen in the future?
Well, some of these sites, actually several of the sites I worked on, are from what are called Oceanic Plateaus. And these plateaus are raised areas of volcanic crust that are out in the middle of the ocean basin. And they've been pretty protected from any kind of tectonic. So like in North America, we had the collision of Africa with North America causing the rise of the Appalachians. And then, of course, you have the Tetons in the west and mountain building episodes on various continents worldwide because of these continental collisions. but these seamounts and oceanic plateaus are just quiet areas that don't suffer from any kind of tectonics, except with time, seafloor spreading is going to subduct them. And so the future of all these plateaus is eventually not very good. They're eventually going to go and meet some subduction zone and disappear from the geologic record. So the Earth is constantly generating new crust and, of course, reabsorbing old crust. And this is just going to go on for millions, billions of years. And all the present-day seafloor is going to be gone at some point.
I believe this question is a question in responding to your answer to the previous question. It's dealing with, do you believe that the extinctions only took months to happen? I'm assuming that's related to the question regarding the meteorite.
Right. Yeah. You can imagine how quickly, well, imagine going without food for a few days. I mean, you're going to suffer. So you can last, as long as you have some water, you can last maybe 10 days, right? I don't know how much. How long would a calcareous nanofossil live without food? It gets energy from sunlight. So without sunlight, it can't make its own food. It's going to be a matter of a few days, and they're dead. Benthic Ferminifera, again, you can starve a foram for a week, maybe 2 weeks. But once you lose its food source, it dies. And so all of this did indeed happen over the course of weeks to months. And so it's a very abrupt event.
Okay, and I guess the person wrote in a little bit more, and I guess they were kind of referring specifically to the dinosaur extinction, and that they felt that you had suggested that the mammals might have had survived because of private caches for a couple months. Is that the time scale for dinosaur extinction, just months?
Yeah, it's probably faster than that because, again, if you imagine, well, I don't know, I suppose the Triceratops could forage around for a few weeks on the grasses and things in the dark, but eventually this stuff dies. I mean, just think how long it takes when you throw carpeting on top of the grass. That grass is dead within a week, right? It's not getting any sunlight. So pretty quickly, this food supply is gone for the herbivores, and then all you have are dead carcasses for the carnivores to feed on, and so it's going to be a matter of weeks after that. So I mean, if you were to guess, it's a matter of a few months, and the dinosaurs are gone.
Did you use only one core to produce your Cretaceous curve, or did you use many, and which ones did you use?
Very good question. I should have pointed that out. That curve is based on several of the best preserved sites, including the sites that are on Blake Plateau, sites 1049 and 1050, as well as another one called 1052, all off of northern Florida. Another part of the curve is from Falcon Plateau in the South Atlantic, 58 degrees south. And then another part of the curve is from Maud Rise near Antarctica, the deep benthics from there. And then another part is from the Indian Ocean. And then a fourth part is from a site in the eastern North Atlantic, site 551. So there's probably about 5 sites in the Cretaceous curve that I presented there.
You may have already answered this, but how do you tell different kinds of forams apart?
I did actually answer that. That's a long answer. I could probably just go back to a few slides. Am I still, I guess I'm still being video broadcast.
Yes,
yes.
Still have control.
This slide actually shows a Ligocene species, and unfortunately the Ligocene is really boring because they all look like golf balls, and maybe to most people the plankton forams all look the same no matter what. But basically I see here this species is called Globigerinotheca index. It's got this big aperture here. It is very spherical, This is a chamber, there's a chamber, that's another chamber. But overall, its overall shell is very spherical. Now compare that to this one, which has got sort of a squashed final chamber and 3 other chambers, not quite as spherical. And I can see there's another, you can see this one here. This is the last chamber, second last chamber, third to last chamber, and it coils like this. That's a completely different genus from these other ones. So, and then here's a 4th species. So that's an example of for the Oligocene. I could show another one. Well, we'll show this slide. These are the modern plankton forams that I showed before. So each of these is a different species. And you can see like this one has a keel all the way around the outside. A keel is a thickening of the shell. And you can see the coiling is much more open and it's kind of flatter compared to this. These are much more spherical chambers. This one has very distinct spines coming off, a very smooth shell. So each of these has distinctive characters. This one has got what's called a bulla. This one has multiple sutural apertures. So there's all these morphology terms we have for these chambers and the apertures and the sutures between the chambers, all that stuff. So there's a lot of details about the way the chambers form, the position of the aperture, the wall texture, or how bumpy it is, if it's spiny, if there's large holes or small holes, things like that. So we use a lot of different features to identify to the species level.
And I think we have time for one last question. Have you considered in the near future to complement your isotope data with other geochemical analysis such as sulfur?
Well, sulfur isotopes give you something completely different, but in fact we do something for oxygen isotope temperatures to sort of determine whether or not we have multiple indications of a warming event or a cooling event. One tool is called magnesium-calcium ratios, and those ratios are directly proportional to the temperature if you have the right kind of shell preservation. And so by measuring oxygen isotopes together with magnesium-calcium ratios, if those curves show parallel trends, then you know it's a temperature-driven curve. If you show that the oxygen isotope curve has a different pattern than the magnesium-calcium ratio curve, that would suggest that maybe you have a salinity effect or an ice cap effect influencing the isotopic composition of the ocean water. Another tool is called TEX86. This is an organic biomarker, which is a pretty new tool, but you have to have a special kind of organic molecule called from something called crenarchaeota, which do not occur in all sediments. However, there are places and opportunities to use TEX86, and it's been quite useful for a number of studies. So we're constantly looking for additional tools to reconstruct the oceanography and sort of chemical history of the ocean. Nitrogen isotopes, sulfur isotopes, there's a lot of different things that we use to interpret productivity and a lot of other factors. So yeah, this is sort of the golden age for oceanography because we've got so much good instrumentation and such improved knowledge of these geochemical markers.
Okay, well I think we're probably out of time for, we have a couple last questions. So we'll send this to you. If you're able to answer them, we'll try to post them online for everybody.
Okay.
Well, I am actually going to take control back of the screen for 1 second. On behalf of everybody who participated tonight, I'd like to thank Dr. Huber for his presentation. He did a fantastic job, and I'd just like you to know, Dr. Huber, that quite a few people have written in and said thank you. They were very happy to participate tonight, and we even had one say that they're very excited to hear about all this because they're teaching plate tectonics right now in school. So again, on behalf of all of us, thank you.
Okay,
and
good.
For those of you with us, just one more reminder. if you go to the NOSB.org website under professional development tomorrow morning, as soon as I can, I will get Dr. Huber's presentation posted along with any resources that he might send us. And just to give you a brief heads up, our next webinar is going to be held on December 19th. I'm not 100% sure if it's going to be at 6 p.m. or 8 p.m. yet, but I will definitely let everyone know next week. I'll send out the invitation. And that one is going to be with Mr. Walter Musial, and he is with the National Renewable Energy Laboratory. And we are still planning to postpone the presentation on phylogenetic trees and jellyfish, and we're hoping to hold that sometime in early January. So again, be on the lookout from 2 more emails from NOSB with invitations to join for those webinars as well. So once again, thank you everyone for participating tonight. We ended right on time, which is fantastic. And again, thank you, Dr. Huber.
You're welcome.